talk about the cellular respiration. Well, it's actually three processes combined together to generate the energy from the glucose molecule, and they are the glycolysis. This occurs in the cell cytoplasm. Krebs cycles. This occurs in mitochondria matrix, and electron transport change in mitochondria in the membrane space. All these are important because our cells need energy to work. But in this video, I only talk about the glycolysis process. The term glycolysis is coming from the Greek words, which glykos meaning glucose, and lysis meaning breakdown. When combined together, it becomes the glucose breakdown. Simply said, that's how it's explained. Now, glycolysis is also a sequence of reaction with enzymes which converts one glucose to two pyruvate molecules with the production of ATP, the adenosine triphosphate, the energy. And glycolysis is the major pathway in the tissues lacking mitochondria, such as the red blood cells. Our red blood cells have no mitochondria, so in order to obtain energy, glycolysis is used. Now, imagine this is our cells. In cells, there are many membrane-bounded organelles, and we can find the enzymes in the cytoplasm. These are the 10 enzymes involved in glycolysis. Hexokinase, the phosphoglucose isomerase, the phosphofructokinase, the aldolase, triose phosphate isomerase, the GAPDH, the phosphoglycerate kinase, the phosphoglycerate mutase, enolase, and pyruvate kinase. They're cute, isn't it? Now this is glucose. The exokinase will take the phosphate from the ATP and bind it to the 6 carbon of glucose, forming glucose 6-phosphate. Then, phosphoglucose isomers change the structure. Fructose 6-phosphate form. Next, the phosphofructokinase will take the phosphate from the ATP and bind it to the first carbon, and fructose 1,6-phosphate form. Then, aldolase will cut the molecules into 2 molecules. DHAP and GAP form. Next, the triose phosphate isomerase change the DHAP into GAP molecule. 2 GAP molecules form. Next, the GAPDH enzyme then will add the phosphate from cytoplasm and bind it to the first carbon of each molecule. The GAPDH work 2 ways, where the NAD plus come and take the hydrogen from the molecules, forming the NADH molecules, and 2 molecules of 1,3-bisphosphoglycerate form. Now, the phosphoglycerate kinase break down the bond of phosphate and the first carbon of each molecule. It releases 2 molecules of ATP and producing 2,3-phosphoglycerate. Phosphoglycerate mutase bind each phosphate at the second carbon of each molecule. 2 phosphoglycerate form. Next, the enolase enzymes take the water from the molecules, producing 2 molecules of PEP. Lastly, pyruvate kinase remove each phosphate from the molecules and bind it to each ADP molecule. Then, 2 molecules of ATP release and 2 pyruvate form. And the product of this glycolysis are 2 pyruvate, 2 NADH, and 2 ATP. And that's how the glycolysis process works. Now, the 2 pyruvate need to go to their new journey, which is the Krebs cycle. And that's all from me. Thank you.